Mendel's experiment showed that offspring inherited one of two versions of a given trait. These traits were inherited by offspring receiving two copies of each characteristic, one from each parent, whether you like it or not. And so meiosis wasn't understood until after Mendel's time, but we now know that traits are housed on chromosomes, and since we inherit a set from mom and a set from dad, we have two copies of each trait. However, meiosis leads to haploid gamete formation, which only has one copy of each trait. Thus, the union of two separate gametes leads to offspring with copies from both parents. Mendel called these copies factors, but it was actually Charles Darwin that originated the term that we use called genes. For all the genius behind Darwin discovering the mechanisms of evolution, he failed to provide an explanation for genetics experimentally. He did, however, hypothesize an idea called pangenesis. In it, all cells of the body produce tiny gemmules, which contain hereditary components. Upon the formation of gametes, these gemmules fuse to the sex cells and the offspring's traits are determined at fertilization. We now know that meiosis is the driving factor of gene diversity in the body, and that fertilization, where the union of gametes, provides the potential offspring with their heritable traits. Still, the word gene can be attributed to Charles Darwin. I'll tell you what's sad is Darwin and Mendel existed at the same time, but they never met. If they did, we might have flying cars, human deep space exploration, or even ice cream machines that work at McDonald's. Alleles are variations of a genetic trait. Purple and white flowers are variations for floral color in pea plants. Thus, there is a purple allele and a white allele. And while Mendel only discovered two alleles per trait within pea plants, it is possible that some genes have three or more alleles. The two alleles that make up an organism's traits describe that organism's genotype. The physical expression of that genotype would be the organism's phenotype. In terms of genotypes, if one has two of the same type of allele, they are described as homozygous. If they have two dominant alleles, they are homozygous dominant. If they have two recessive alleles, they are homozygous recessive. If an individual has a dominant and a recessive allele, their genotype is heterozygous. In the case of Mendel's pea plants, using flower color, the phenotype of purple flowers will result in a genotype that is either homozygous dominant or heterozygous. If the phenotype is white flowered, the genotype is only homozygous recessive. Mendel always used purebred plants in the parental generation. Therefore, these pea plants were either homozygous dominant or homozygous recessive. Enough about pea plants, let's apply this to humans. A small segment of the human population has inherited phenylketonuria, a genetic disorder that makes it difficult to metabolize food high in the amino acid phenylalanine. And you may have noticed that on the back of a diet soda, there is a warning that says phenylketonuric contains phenylalanine. If you have phenylketonuria, you are homozygous recessive. If you don't, you are either homozygous dominant or heterozygous. If heterozygous, you don't have phenylketonuria, but you do risk passing it on to your offspring. If your partner is also a carrier. However, if you are diagnosed with phenylketonuria, it is treatable by having a diet low in phenylalanine. Going back to peas, yeah, I know, kind of boring. But why was it that when Mendel used purebred or homozygous plants, the F1 generation always displayed the dominant phenotype? Well, this is because of the law of dominance, where one allele masks the expression of the recessive alleles. In flower color, the homozygous dominant purple-flowered pea plants and homozygous recessive white-flowered pea plant always produce purple-flowered offspring, but all of these offspring are heterozygous. That is, they receive a purple allele and a white allele. But since white is recessive, it is masked by the dominance of the purple flower. When members of F1 self-pollinate, around 75% of the offspring will have purple flowers and 25% of the offspring will have white flowers, or a 3-to-1 ratio! And this is because it takes two recessive alleles to express white petals. In fact, the only way a recessive phenotype can be expressed is if the genotype is homozygous recessive. As mentioned earlier, the F2 generation of all of Mendel's pea plants had a 3-to-1 dominant to recessive ratio, and this was determined by the use of monohybrid crosses, or a cross using one trait at a time. Performing monohybrid crosses is a form of probability, which is the mathematical likelihood that something occurs. A probability of 1, or 100%, means an event will be guaranteed to occur. 0.5 or 50% means equal chance of occurring or not occurring. And a probability of 0 means a 0% chance of occurring. If you look at pea pod color, green is dominant and yellow is recessive. If the parental generation is purebred, the F1 generation is then green but heterozygous. If self-fertilization occurs, the F2 generation can be predicted using a probability tool called a Punnett square, named for British geneticist Reginald Punnett. Parental genotypes are placed on the top and side of the Punnett square, and no, it doesn't matter who's on top. Note that the lines separating the genotypes into alleles represent the haploid sex cells. Each sex cell carries only one copy of each gene. The 4 offspring genotypes represent the probability of each diploid genotype possible with these 2 parents. This creates both genotypic and phenotypic ratios. In this case, the self-fertilization of the F1 generation will yield a 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive genotypic ratio. And, believe it or not, a 3-to-1 ratio! The ability to predict probabilities in the offspring made it easier to control for specific traits wanted in an agricultural product. For example, while yellow peas are dominant to green peas, they also have a milder flavor. Green peas are sweeter and sell better, so to make green peas, ensure that only green pea plants cross. Due to a 25% chance of a homozygous recessive phenotype showing up in the F2 generation, Mendel proposed that there were 2 genes, or factors as he would call them, per trait. Without the aid of microscopes or cell biology, Mendel predicted that during gamete formation, these alleles in a parent's genotype would separate, and that during fertilization, the parental genotypes fused, making a zygote with its own genotype. Mendel called this the law of segregation. And we now know that when cells undergo meiosis, the mother cell, which is diploid, having 2 copies of each chromosome, which is housed in genes, will produce 4 haploid egg or sperm cells with only 1 chromosome copy each. In other words, each gamete has only 1 allele per trait. The genotype won't be determined until both the sperm and egg unite to make a zygote. In Mendel's experiments, those that displayed the recessive phenotype were always homozygous recessive in their genotypes. However, if a pea plant had a dominant phenotype, how would you know if the genotype was homozygous dominant or heterozygous? Well, to do this, one would perform a test cross where one parent displays the recessive phenotype and the other displays the dominant phenotype. In humans, the ability to taste the molecule phenolphthaleurea is considered dominant, and the inability to taste it is recessive. Now, please keep in mind that this is oversimplified and more complex, but still a fun trait to try out. I am a non-taster, so if we were to use the letter T, my genotype would be 2 little t's. My wife is a taster, but I'm unsure if she is big T, little t, or 2 big T's. Therefore, our genotypic and phenotypic ratios could go in one of 2 ways. If she is homozygous dominant, then all of her offspring are heterozygous, or tasters. If she's heterozygous, then there's a 50% chance that the child is a taster and a 50% chance that the child is a non-taster. Come to find out, I have one child who's a taster and another who is not. Therefore, my wife's genotype is heterozygous for PTC tasting. So what is the science behind PTC tasting? Well, we still don't know, but the chemical composition of PTC is similar to the dislike of plants in the mustard family, such as broccoli, Brussels sprouts, and cabbage. Therefore, a person with strong PTC taste might be averted to eating these foods. Others speculate that this gene may have helped many of our ancestors avoid toxic plants altogether. Mendel used dihybrid crosses as well, which are matings between parents that are both heterozygous for 2 traits. And to determine whether the principle of segregation holds true if parents differ in more than 1 trait, Mendel's results supported the principle of independent assortment, which states that alleles of different genes are transmitted independently of one another. We now know this is the case because genes can be housed on differing chromosomes. So to demonstrate, let's take a purebred purple pea plant and a green potted pea plant with a purebred white flowered yellow potted pea plant. We should expect all of the offspring to be purple flowered and green potted, but heterozygous for each. So from there, We can build a dihybrid Punnett square by breeding the two plants of the F1 generation. Before beginning, we have to determine the different combinations of gametes that each plant can make. In this case, parents can make the following gamete combinations, since each gamete can carry only one allele per gene. These combinations are big F and big G, big F, little g and big F, and little f, little g. Afterwards, the offspring will have the following genotypes by placing the potential genotypes together. The Punnett square that results from a dihybrid cross predicts that there should be 9 different offspring genotypes and 4 different phenotypes. The 4 possible phenotypes should be presented as a ratio of 9 to 3 to 3 to 1. We abbreviate dominant genotypes with the first letter capitalized and the second letter as a dash, since it doesn't really matter if the second allele is dominant or recessive, the phenotype is the same. We now know that independent assortment takes place during meiosis 1, specifically metaphase 1, as the chromosomes line up on the metaphase plate. This lining up is random with each chromosome, as is in periphase 1. A variation of both maternal and paternal chromosomes occurred by crossing over. Genetics, however, does not always follow the patterns that Gregor Mendel discovered. So in our next video, we will be looking at other modes of inheritance that exist.